All right, so this is the second video, again talking about how the body makes sex cells or gametes, and just something that's a little bit more particular to us. So when we're looking at humans, what we see is that in human males, prophase is going to last about 1 week. So thinking about the phases of mitosis, prophase 1 lasts about a week. In males. So just interesting. Human females, prophase 1 actually begins before you are born. And that meiosis will continue up to the point where you no longer are going to produce any more eggs when you go into menopause. So, but the meiosis doesn't continue until after you've ovulated and that egg has actually been fertilized. So interesting. But getting back to this idea of, well, how do we get more diversity in populations as a result of sexual reproduction, in this case, the production of gametes or sex cells? What we see is that this is one way. One way is that during meiosis 1, we get this exchange of information from the parent who's making the sex cells, right? So they inherited some chromosome number 1 from mom and number 1 from dad. Each of them has made their copies. Now we're producing these gametes, so we have this little exchange that's happening. So we're getting new combinations of that information. But there's also another way that a lot of diversity happens, and that is something called independent assortment. So during anaphase 1, it's truly a matter of chance which of the sex cells ultimately is going to get which of the combinations of these sister chromatids, these homologous pairs. And you can see there's just a tremendous amount of potential for diversity. So in humans, remember that we have 23 pairs of chromosomes. So statistically, you could say 2 to the 23rd, which is this big number here. Different combinations between the maternal and the paternal chromosomes can be produced. So let's take a look. So this is ignoring the idea of crossover now, but that happens too. But let's just ignore that for now and say, okay, these original cells that are going to make gametes that you might have in you, you got a pair. Sorry. Got that lovely chime in the background. You got one pair from your mother. Here you got a pair from your father. Each one of them has made copies. So here you can see now we have one from the homologous pairs from mom and dad with their sisters that are all lined up here at metaphase 1. And then in telophase 1, there's no telling which way this is going to end up, because look, you had 2 options. You could have gotten on this side, if we call the red ones from mom, we could say that the mother's chromosomes and their sisters have all lined up here on the left of this equatorial plate, this middle point, and then the fathers are here. So that could be one possibility, in which case this is what that first telophase set of cells would look like. But there's nothing to be said for it not happening like this, where the way they've lined up during metaphase 1 results in that first division giving you these possible, one from mom, one from dad, one from mom, one from dad. So it just adds to a lot of potential differences between the gametes. Now, something that also can happen, and we talked about this a little bit in the cell cycle, especially in metaphase, is what happens if there's a failure to separate the sister chromatids at anaphase 1 or anaphase 2. That's going to result in something called aneuploidy, which means sort of without. Aneuploidy means that you're going to have an abnormal number of chromosomes in some of those cells. So let's take a look at what that would be. So here's meiosis, and you can see what's happening here is that we have this, in some cases, we're going to get extra chromosomes, and in this case, this is just following one pair of chromosomes. So if this cell didn't receive any, and this got both of them by accident, then when that cell gets fertilized by the sperm here, then what you're going to end up with is that the end, you're going to get just a single chromosome where you should have had a pair. And on the other side, when you didn't separate, you ended up with the pair plus the one from the sperm, so you end up with something called trisomy. And this is monosomy. And you can imagine without an entire chromosome here, that's not likely to, that baby, if that would not develop. In other words, it would be a spontaneous abortion or miscarriage. Same here, could be, in some cases, fatal, wouldn't develop, although there are some exceptions. And that is here. So trisomy 16 is one that can happen, as well as trisomy 21, which is what we commonly know as Down syndrome. Polyploidy is really interesting because polyploidy means you get multiple sets. So instead of being 2N, an organism can be 3N or 4N or even up to 8N. So down here what you see is that happens a lot in plants. And sometimes it happens because of how we try to not genetically modify, but how we develop certain, in this case, strawberries that have a particular quality, like their size. So this can occur through extra round of DNA replication or something. It can be due to mistakes, but there are a lot of polyploid plants. So we're going to actually use strawberries, I hope, this week and try to extract some DNA. And they're a good source of DNA because they have this polyploidy condition, this octoploid with 8 chromosomes in them, or sets. So we'll go a little further here. So all of that is to say, between the crossing over event that can happen with the gametes and the independent assortment, what we end up with are just a huge number of possibilities that contribute to this idea of variation or diversity between species and within species. So we can see all sorts of interesting combos and effects in our world. We can also look at ourselves in our family trees. So this is called a pedigree. And the pedigree is telling us that here there was a female that was carrying, in this case, the disorder, the gene for hemophilia, but only had one copy of that gene. And here's the normal male, and so these are like the grandparents. And they had kids, and you can see by following the line directly, you can see who their kids were. So here's a male that inherited hemophilia. There's a male that is normal. There's a female that is normal. And here's a female which ends up being a carrier for that gene for hemophilia, that then she marries this normal male, and you can just follow the lineage. So how many of her offspring are normal versus carrier, because it's a female, versus a male that has hemophilia, because he has this is a sex-linked trait. So we'll talk about this later on as how that works. But it's really fascinating to look at family history and kind of trace certain traits, not always disease traits, but certain traits over multiple generations. And that's what this pedigree is. Now, a lot of our understanding of genetics it really comes from this monk by the name of Gregor Mendel, who lived back here in this time period, considered the father of modern genetics. Just a really observant person who tended the garden and did all sorts of, he spent a lot of time outdoors. And when he eventually went into the abbey, and he kind of conducted some experiments that had never been really thought of before. He was just an observer and watching how certain plants seemed to produce some similar or variations in those traits. So what he looked at in particular, what he's famous for, is studying peas. And pea plants were easy to study because they had very distinct what are called dichotomous traits, meaning that their flower color would be sort of an either one color or another. Their seed shape would be one color or another. Seed color itself could be one or the other. So it's an either or kind of thing. And what he could do is see when those traits occurred and which plants actually caused had sort of purebred offspring, and which ones showed some variation. You could manipulate how these plants actually got fertilized, because in a single plant, you have both male and female sex organs in that same plant or in that same flower. So you could use that to sort of manipulate how it got pollinated. This is called Meiosis. The other reason they're good to use is because they're really easy to get. They're cheap. They reproduce very quickly at a short generation time. So that's why he chose to use peas, because peas can self-fertilize, but you can also remove this pistil, which is, that the pistil is part of the female part of the plant. You can remove any parts of this, like the stamen where the pollen is. You can remove it, or you can use it to pollinate some other plant next door. So kind of handy to do some controlled kind of experiments. Now, these vocab words are brought up in gizmos, at least some of them. Not all of them are ones that we're going to focus on, but we're going to look at things like the parental generation and the first filial, which is the first offspring from those parents, and then what their offspring are, the F2 generation. So we're going to spend really more time looking at that, and then we'll talk a little bit about this idea of true breeding. We're getting kind of close on time, but let's take a look at one. So here's the parents, the P generation, and here we see these pretty purple flowers. And if you had a purple flowered plant and you're going to cross it with another purple flowered plant, the offspring of those two is purple. So no big surprise. Then we take those two and we're going to cross it again with one of its same generation. So that's why they're saying one of its siblings here. And then, then the F2, we don't see any difference, right? We get another purple plant. So this is a true breeding situation where we have a single trait and it's purebred for that trait, and we're going to keep getting that trait over and over multiple generations. But we can also do this, and that is take some plant that's true breeding for purple and take another variety of that pea plant that's true breeding, but in this case for a white flower. When we breed those two together, or when Mendel did rather, he got purple. There's like no white. So it looked like the purple color was dominant and kind of overshadowed the white. However, if you go one more generation, you can see that in a 3-to-1 ratio, we have 3 purple to 1 white. So now the white show up again. And we're going to talk about this in a little bit more detail coming up. So stay tuned.